Lesson 8. DNA. DNA is like a blueprint or instruction manual for living things. It is found in every cell of our bodies and contains all the information that makes us who we are. Imagine DNA as a long twisted ladder or a spiral staircase. This ladder is made up of smaller building blocks called nucleotides, consisting of a backbone of sugar and phosphate groups. Attached to each sugar will be one of 4 chemical bases: adenosine, thymine, cytosine and guanine, represented by the letters A, T, C and G. These nucleotides pair up in a specific way. A always pairs with T and C always pairs with G. So if you have an A on one side of the ladder, there will always be a T on the other side. And if you have a C on one side, there will always be a G on the other. The order of these nucleotides along the DNA ladder is what makes each person unique. It's like a secret code that tells our bodies how to grow, develop and function. This code determines things like our eye colour, hair colour and even our height. DNA is passed down from parents to their children. Half of the person's DNA comes from their mother and the other half comes from their father. This is why children often resemble their parents or other family members. Scientists can study DNA to learn more about different living things. They can use DNA to solve crimes, identify people, and even understand how different species are related to each other. So in simple terms, DNA is like a special code that tells our bodies how to work and what we will look like. It's what makes each of us unique and connects us to our family. DNA and forensics. DNA is an incredibly valuable tool in forensic science. It can be used to identify individuals, link suspects to crime scenes, and exonerate innocent people. DNA profiling. Forensic scientists extract DNA from biological samples found at crime scenes, such as blood, saliva, hair follicles, or skin cells. They can then analyse the DNA to create a profile which is a unique genetic fingerprint of an individual. This profile is generated by examining specific regions of the DNA that vary between individuals. Comparing DNA profiles. Once a DNA profile is obtained from a crime scene, it can be compared to DNA profiles in databases, such as those containing profiles of known offenders or missing persons. If a match is found, it can provide strong evidence linking a suspect to the crime. Identifying unknown individuals. In cases where the identity of a victim or suspect is unknown, DNA can be used to establish their identity. By comparing the DNA profile of the unknown individual to profiles in the databases to family members, forensic scientists can determine their identity or even if and how certain suspects are related. Cold case investigations. DNA technology has been instrumental in solving cold cases where the trail has gone cold for years or even decades. By re-examining old evidence and using advanced DNA analysis techniques, investigators can sometimes identify the perpetrator or establish new leads. Exonerating the innocent. DNA analysis has been crucial in proving the innocence, exoneration of individuals who have been wrongfully convicted. By comparing DNA evidence from the crime scene to the DNA profiles of the convicted individuals, innocent people have been proven innocent and released from prison. It's important to note that DNA evidence is highly reliable, but requires careful handling and analysis to ensure accuracy. This is because the DNA can be damaged by chemicals, heat or UV light. Forensic scientists follow strict protocols to prevent contamination and ensure the integrity and stability of the DNA samples. DNA evidence has revolutionised forensic investigations and has played a significant role in solving countless crimes. Blood groups. Blood groups, also known as blood types, are determined by the presence or absence of certain proteins on the surface of red blood cells. The most well-known blood group system is the ABO system, which classifies blood into 4 main types: A, B, AB and O. Here's a breakdown of the different blood types in the ABO system. Blood type A. People with blood type A have A antigens on the surface of their red blood cells and anti-B antibodies in their plasma. This means they can receive blood from donors with blood type A and O, and they can donate blood to recipients with blood types A and AB. Blood type B. People with blood type B have B antigens on the surface of red blood cells and anti-A antibodies in their plasma. This means they can receive blood from donors with blood types B and O and can donate blood to recipients with blood types B and AB. Blood type AB. People with blood type AB have both an A and B antigens on the surface of their red blood cells and no antibodies in their plasma. This means they can receive blood from donors with any blood type, A, B, AB or O, but they can only donate blood to recipients with blood type AB. Blood type O. People with blood type O have neither A nor B antigens on the surface of their red blood cells, but they have both anti-A and anti-B antibodies in their plasma. This means they can only receive blood from donors with a blood type O, and they can donate blood to recipients with any other blood type, A, B, AB or O. Blood type O is the most common blood type. In addition to the ABO system, there is another blood type system called rhesus system. The rhesus system classifies blood as rhesus positive with the presence of rhesus antigen or rhesus negative with the absence of the rhesus antigen. The rhesus factor is important in blood transfusions and during pregnancy, as rhesus negative individuals can develop antibodies against rhesus positive blood if they're exposed to it. Blood group forensics. Blood groups can be used in forensic investigations in several ways. Bloodstain analysis. Forensic scientists can analyse bloodstains at a crime scene to determine the blood type of the individuals involved. This information can help investigators narrow down potential suspects or victims. Blood typing of suspects. If a suspect's blood type matches the blood type found at a crime scene, it can provide evidence linking them to the crime. Conversely, if a suspect's blood type does not match the blood type found at the crime scene, it can help exclude them as a potential suspect. Victim identification. In cases where the identity of the victim is unknown, blood typing can be used to establish their blood type. This information can compare for missing persons records or used to narrow down potential matches. Paternity testing. Blood groups can also be used in paternity testing. By comparing the blood types of a child and potential parents, forensic scientists can determine the likelihood of biological parentage. Mass disaster investigations. In a mass disaster situation, such as plane crashes or natural disasters, blood typing can be used to identify victims, while other forms of identification are not possible. It's important to remember that while blood typing can provide useful preliminary information in forensic investigations, it is not as precise as DNA analysis. DNA analysis can provide more conclusive evidence, as is typically used in conjunction with blood typing for a more comprehensive investigation. Blood detection and patterns. No matter how much you clean up a crime scene, blood is very difficult to erase without some very heavy-duty chemical cleaners. Tiny particles of blood can cling to surfaces or hide in small surface cracks and still be identified as blood years later. Forensic investigators can use certain chemical analysis methods to determine if any blood found at the scene is of human origin or from another source. Luminol works by the process of chemical luminescence. This is a chemical reaction between the hemoglobin in the blood and a combination of chemicals that result in the production of light. Glow sticks work on the same principle. The chemical component of luminol is a powder that is composed of nitrogen, hydrogen, oxygen and carbon. The powder is mixed with a liquid that contains hydrogen peroxide. The iron in the hemoglobin acts as a catalyst for the reaction and speeds it up in a process called oxidation. In this reaction, the luminol gives away some of its nitrogen and oxygen to the hydrogen peroxide and basically exchanges them for some oxygen atoms. This causes a release of energy in the form of light and will make the blood glow if the lights are turned way down low. The Kastle-Meyer test for blood. A similar test to the luminol, but using phenolphthalein with the hydrogen peroxide. This also reacts with the iron in the blood, but instead of fluorescence, it reacts with an enzyme to form a colour change from clear to bright pink. But you have to be pretty quick, because if it is left longer than half an hour, the reaction happens anyway, and the swab will turn pink on its own. The test is actually really sensitive, and one drop of blood diluted in 10,000 drops of water can still be detected. On the downside, some chemicals and other bodily fluids that are not blood can also be detected, and so you have to be careful it is not a false positive. That's why this test is used as a starting point and not a conclusive test. Blood pattern analysis. Also known as blood stain pattern analysis, BPA. It's a forensic discipline that involves the examination and interpretation of blood stains at crime scenes. It can provide valuable information about the events that occurred during a crime scene and help reconstruct the sequence of events. Here are some key aspects of the blood pattern analysis. Types of blood stain patterns. Bloodstains can take various forms depending on the force and direction of the impact and surface on which the blood lands, and other factors. Some common types of bloodstain patterns include passive stains. Bloodstains created by the force of gravity, such as drips, flows and pools. Impact stains. Bloodstains created by a forceful impact, such as splatters, splashes and cast-off patterns, splatters from the weapon after hitting the victim. Transfer stains. Bloodstains created when a wet, bloody surface comes into contact with another surface, leaving a recognisable impression or pattern, like a bloody handprint or footprint. Projected stains. Bloodstains created by a forceful projection of blood, such as arterial spurts or forcibly inhaled, breathed out blood from coughing or sneezing. Analysis and interpretations. Blood pattern analysis examine the size, shape, distribution and other characteristics of bloodstains to determine how they were created. They consider factors such as the angle of the impact, the velocity of the blood, and the distance between the source of the blood and the target surface. By analysing these patterns, analysis can provide insights into the dynamics of the crime scene, such as the position and movement of the victim and the assailant. Reconstruction of events. Blood pattern analysis can reconstruct the sequence of events during a crime. By analysing the location and pattern of the bloodstains, analysis can determine the relative positions of the individuals involved, the type of weapons used, the number of blows or shots fired, and other important details. This information can be crucial in corroborating or challenging witness statements. Bloodstain pattern documentation. Blood pattern analysis carefully document and photograph bloodstain patterns at a crime scene. They use specialised techniques such as stringing or laser trajectory analysis to determine the origin and direction of the bloodstains. This documentation is important for later analysis and presentation in court. It's important to know that blood pattern analysis is a complex field that requires specialised training and expertise. Forensic scientists who specialise in bloodstain pattern analysis undergo intensive training to accurately interpret bloodstain patterns and provide expert testimony in criminal investigation and court proceedings.